they'll play kind of,
Yeah. Same for me. Yeah.
Yeah.
Do you have that that option to optimize for video sharing?
I think I did that.
What do you mean? What is optimized for video?
So when you click share screen at the bottom left corner, there's an option that you can click.
Oh, yeah,
and then it'll optimize.
I have it.
And sometimes it's grayed out for me and sometimes it's not, and I don't know why. It does require a minimum, internet connection to be, certain upload speed.
But if you reversible once you click it, can you unclick it during the talk?
Not during the talk, but it doesn't change anything. The only thing it changes is it makes it so you can't see the audience. Like the video previews of all the faces, it'll cut that off and there's no way, I think, to see those. So if that matters to you, but most people keep their cameras off anyway.
Right.
Well, I'm a teacher as well, and usually I don't like it when I don't see any students.
Well, people, we can always request that people keep their cameras
that's
on.
what I do usually, but prevents you from having the feeling of talking to your computer.
Yeah, no, I was actually in an interview and all of a sudden this fear came over me because I didn't have any feedback or anything that I was talking to myself. And I just stopped and I was like, are you guys still there? Right. Is anyone there? And they had to reassure me because it was just this overwhelming certainty that I was talking into the void. And luckily, they were still there. It was fine, but it's deeply strange. Yeah.
Like you didn't realize your internet connection has failed like 10 minutes ago.
Right, because you wouldn't know.
Yeah, right. How would you exactly? Yeah.
You could just be talking to no one and you wouldn't know. Sounds like a Twilight Zone episode. Also, we didn't check with you guys ahead of time. Anyone prefer to go first?
I don't care either way.
Okay. So you're both equal time. We usually have the shorter talk go first, but since we're 50-50 here, I think actually.
Yeah, Bob, go if you want.
Okay, sure. Yeah. Yep, I can go first.
All right. Well, let's go ahead and get started. And I think Adam.
Yeah, we're good. You're on YouTube, right?
Yep. Streaming there. It's all good.
Okay, good. So let's get started. So hi, everyone. We've got 2 speakers today. Just a reminder that if you have any questions for the speaker, you can use the raise hand feature on Zoom, or you can type your question in the chat, or if you're on YouTube, also type your question in the chat. So our first speaker is Bob Fischer. Bob is a staff scientist in Claire Waterman's lab at the NIH. He did his PhD studying viral oncogenes and then postdoced with Velia Fowler working on actin capping proteins at the Scripps Research Institute in La Jolla, where he met Claire and then came to the NIH with her in 2008. He teaches on the MBL physiology course since 2009, and today he's going to be talking about how waviness of ECM fibers affects cancer polarization and migration. So take it away.
Great, thank you. Let me try and share my screen here, and I'll click the optimize. Okay. Let me see and go to. Are we clicking? Okay. So you guys are seeing this. Let me start off by thanking the organizers for giving me an opportunity to talk in this excellent seminar series on some work that I've been doing in Claire Waterman's group for the past few years, and we hope that it will be coming out very soon. We're in the sort of last round of minor revisions there. And by way of thanking the organizers and sucking up a little bit, let me start off by pointing everyone to this excellent review that was written by Adam Schellert and Roberto Mayer. To make my first point, which is that cells are able to sense a wide range of external cues. And when these cues are arranged in an anisotropic or polarized way, the cells can use these cues to polarize. And so Adam's review covers many, many more types than what's shown here. But chemotaxis, durotaxis, haptotaxis all arise due to spatial differences in soluble factors in matrix stiffness and cell adhesion, respectively. So while it seems like this might be a little bit of a bringing coals to Newcastle for this audience, I just want to make the point that directional cell movement requires recruitment of some common cellular machineries in a spatially organized manner. And it's the spatial organization that they'll really be focusing on later. So to initiate cell migration, cells have to generate a forward protrusion of some sort. And this is often typically by actin polymerization, usually by activating nucleating factors such as ARP23 and formins, and or maybe elongation factors like MENA and VASP. Then the cell has to adhere to the substrate, at least for adherent cells. And so cell adhesion, it turns out, is very tightly coupled to that actin-based polymerization machinery, as you can see here in the movie on the right, where nascent adhesions are forming during the actin polymerization-based protrusion. And so they're happening as that happens, so it's coincident with that polymerization. And so then retrograde flow and retraction from this actin network can then apply force to these adhesions and strengthen them to allow them to become more mature focal adhesions. Then finally, the cells can, the organization of the movement can be further enhanced by myosin activity. And this can be done in a couple different ways. So myosin can assemble into contraction fields like you're seeing here in the movie on the right in the lamella region. And these contraction fields flow retrograde to the direction of the cell motility, where in here we're seeing GFP myosin 2A labeled in a cell that would be crawling upwards. So in addition to these arc-like structures, the myosin can assemble into stress fibers. And so these two types of actin-myosin contractile apparatuses can then exert forces via the adhesions to pull the cell forward. Lastly, adhesions then must be disassembled or detached in some way for the cell to move forward. And so again, the important point to raise in this, bringing up this basic model, is to say that all of this must be done with some spatial control. And it is often the adhesions and the polymerization machinery that really set up the directionality of the cell and the organization of these apparatuses within the cell. So cancer cells make use of all of these types of polarization cues, these anisotropic cues, to leave the tumor and migrate around the body during metastasis. And so chemotaxis has been pretty well described by a number of groups, but one of the ones I really like to point to is this sort of work by Bob Insel, where he describes how melanoma cells can degrade LPA in the center of a tumor, leaving an outward-facing gradient for the cells to crawl up. Another example that's been sort of well documented now by John Condilis's group and others is the case in breast cancer. Mammary tumor cells and the local macrophages create sort of a paracrine loop where the tumor cell shown here in purple is creating colony stimulating factor 1. It's inducing the macrophage to be activated and migrate, which then causes the macrophage to produce EGF and causes the tumor cell to migrate. And then earlier in this series, we were treated to some of the seminal work by Madeline's group shown here, who has done some, and she's gone on to have her own lab and done some great work showing that mammary tumors not only change the constituents of the ECM to encourage cell migration, but they also set up spatial differences in those ECM components so that fibronectin and collagen 6 can form gradients leading cells out of the tumor, which is essentially, which is haplotaxis. And then finally, while the notion that more aggressive breast tumors demonstrate matrix remodeling, and that coincides with increased stiffness, and this has been well documented by Patricia Keeley and Val Weaver and others. Recently, Ken Yamada's group showed in a survey of cancer cell types that robust durotaxis is common amongst the more aggressive tumor cell types. But for today, in addition to these types of migration, I want to consider the fact that there is another type of guidance that cancer cells might make use of. And that is the organization of the matrix itself, or the topography of the matrix itself. So we've known for more than 6 decades that cells placed on substrates with linear topographies will migrate preferentially along these shapes. So shown here in the upper left with Curtis and Vard showing that for groups of cells migrating out of a chunk of tissue placed on such a substrate, cells will preferentially crawl along the topography of those substrates. And then later, Andrew Doyle and other people have followed up on this and used this sort of topography sensing to study the aspects of sort of dimensionality of cell migration. And then lately, and as was shown again earlier in an earlier talk in this series, the NAIN group has been using sort of these suspended fibers to further study this phenomenon, which is again called either topotaxis more modernly or contact guidance more classically. But this then goes into somewhat contradiction, or it's sort of in opposition to something that we've known also for many, many years. So for more than a century, we've actually known that matrices in the body often display wavy architectures. And this can be due to the co-expression of things like elastin, which can create sort of cause the collagen fibers to adopt this sort of wavy appearance. But it's also due to the effects of local fibroblasts in mesenchymal cells. And so these examples exist throughout the body. They're common in a lot of vascular tissues. So in the aorta wall or the adventitia of a lot of blood vessels, you will see these collagen fibers that are wavy. And you can imagine that those might be enabling the tissue to expand and contract because they might act as springs. But it should be pointed out that there's other tissue types that also display these very sort of wavy bundles. So you can see an adrenal mesenchyme here in the bottom left, where they get these sort of zigzaggy type collagen fibers. And then in some work that Paul Campagnolo's group has done, really beautiful work showing that in the prostate, using second harmonic imaging here, that the collagen bundles are wavy in addition. So we, from the landmark work of Patricia Caillé and others, we know that many tumors exhibit during their progression a reorganization and remodeling of their ECM architecture. And so as tumors progress in the benign state, they start off, as you can see here, I don't know if you see my pointer, in the benign state, so if the tumor would be here, in these benign tumors, the architecture is very wavy around those tumors. But then as the tumors progress to a more aggressive and more advanced states, the collagen bundles become progressively linearized, and then eventually they become radially arrayed, where they might be acting like roads leading straight out of the tumor. And so this linearization is also seen in other tumor types as well. I'm just showing an example of normal alveolar structure here, and then the metastatic tumor in the lung there, where you can see the collagen fibers are again straightened relative to the normal tumor, or normal tissue rather. Excuse me? Is there a question? Oh, no, maybe not. Okay. Yeah, and I can't see anybody's hand raising, so somebody just please speak up if there's a question. I'm happy to stop and pause. And so there's another example here just shown in the bottom right of head and neck squamous cell carcinoma, where again, you see these collagen fibers become elongated and straightened. So in our own group, we have been looking at developing a mouse model for melanoma. And when our talented graduate student, Michelle Baird, takes a look at the collagen surrounding these tumors, we see that the tumor is again surrounded by these wavy collagen bundles. And as we slice through the center of this tumor, where you see the tumor cell nuclei labeled in purple, we see a lot more linear collagen bundles. And so if I take a Z projection of this and encode the slices with color by their Z depth, you can see, I hope you can appreciate that a lot of the fibers really tend to be kind of one color, which is showing you that they're wavy in a lateral direction rather than being sort of corkscrew type structures. And so if we take a slice to the center of the tumor, again with Z color encoding, and we look at sort of comparing the center of the tumor shown here in the dashed line, you can see again that the center of the tumor has these sort of very linear fibers, collagen bundles, and the tumor periphery is really dominated by these wavy collagen bundles. And if we do quantification of the local curvature along each of these collagen bundles, we see that this is borne out by this quantification, that the periphery of the tumor is really sort of dominated by these wavy collagen bundles. So this then leads us to some questions immediately then. So one is, does the ECM fibril waviness itself alter the cell polarization or directed migration independent of the mechanical properties? So again, you can imagine that these wavy collagen bundles might be and probably have a different mechanical properties, and that would affect the cell migration. But independent of that, just the shape of them, does that shape itself also contribute to the cell behavior? And so if so, then what parameters of that ECM fibril shape might control cell polarization? And then finally, how would the cell respond to this? So how do cellular components allow the ECM fibril waves to regulate cell polarity and motility? So large wave fibers correlate with lower metastatic stroma in breast cancer in a couple different types. So to quantify differences in tumor stroma architecture, I chose to look at these two types of tumors. And so as Madeline pointed out in her talk a few weeks ago, it has been shown that obesity is a is a risk factor for increased breast cancer metastasis and poor clinical outcomes. And so we collaborated with Claudia Fischbach, who shared with us some of her primary tumor data, where she looks at either lean, shown on the top, or obese mouse mammary stroma. And so again, I'm color coding the data here for Z, and you can see again that these fibers tend to be a single color and sort of wavy in the lateral direction. I also used some published data from Paul Campagnolo's group, whose work on ovarian cancer and collagen structure. It was really the inspiration for the work that I'm talking about today. And I highly recommend you checking out some of his work as well for further info. So when we measure then the wave structure for these different types in both these systems, the wave properties, what we find is that the larger waves tend to correspond with more benign behavior or less aggressive stroma, and the more aggressive behavior in these sort of higher grade tumors and in the obese mammary tissue correspond with smaller waves or straighter bundles. And then I would just point out that again, what I'm showing you here in this central column is the measurements of our own mammary, or skin tumors where we look at the collagen structure. And again, it's similar in the order of magnitude of wavelength and amplitude that we see in these other tumor types. So to get this question of does wavy ECM fibril waviness alter cell polarization or directed migration independent of the mechanical properties, what we need to do is have some way of defining the wave shapes such that we can mimic these benign or metastasis-prone ECM fibrils. But then keep all the other cell inputs constant. So we'd like to keep the growth factors and the amount of matrix deposited and the mechanical properties all constant and just vary the shape of the fibrils. So to do this, we collaborated with Zhao Yusan and John Forkas, who have been making these nanotopographies for quite some time to look at cell behavior. And so their nanotopographies these nanotopographic ridges that these guys make can be made with the approximate size of a collagen bundle. And so what they can do then is place many such patterns on one single cover slip so we can do nice side-by-side comparison. So we started with three basic designs. One sort of quote-unquote large wave structure to mimic the more benign stroma that we were looking at in the previous slides. Then a small wave structure that was going to mimic the more aggressive types of stroma. And then we were going to compare that with straight ridges that many other people have been studying and might be representative of the Patricia Keeley tax signature number 3. And so as you can see in this 3D STED reconstruction, we're able to coat these substrates quite well with fibronectin, shown here in blue. And then you see there in green, the paxillin is really predominantly localized along the sides of these fibers and sometimes along the sides of these ridges and sometimes along the tops. Now that isn't to say that there won't be adhesions that can form in between them on the bottom of the cover slip, but if we keep the spacing tight enough, the cells tend not to do that, and they tend to more grab onto the tops of these ridges. So when we then look at how the cells behave on these different types of substrates, well, we can start off to get just a rough measure of polarization. We can just look at the cell shape or ellipticity. And so on both the straight on the top and the small waves, we see that the cells elongate, and whereas on the they're elongate in the direction that they would presumably migrate, and I'll show you that in a second. But then on the large wave substrates, we see that the cells are less elongated. And as another sort of proxy for cell polarity, we can also measure the sort of organization of the internal organelles. And so we look at the organization of the Golgi and nuclear sort of axis that they set up. And what we see here is that in the straight and small waves, they sort of tend to organize their Golgi nuclear axis along the axis of the propagation of the pattern, where in the case of the large wave, which represented those sort of less aggressive matrices, this axis is much more randomized. So the cells are presumably less polarized. And this difference in polarization is borne out by their migration persistence on these different substrates. And so cells on the straight and small waves tend to be more directionally persistent than those on the large wave substrates. But now we get to this leads us to the question, what does large mean? What qualifies as large? What aspects of the large wave depolarizes cells? And so we measured varied several different properties of the wave patterns, but I will just cut to the chase and say that the best sort of property or parameter that we found that was most predictive was the amplitude. So when we look at in this series of shown on the top here, this series of increasing amplitude, but at a constant wavelength, you see that once the amplitude approaches around 15 microns, which is around the size of the larger waves you were seeing in those previous slides, the cells become less organized along the sort of axis of the wave pattern. And so they become less elongated right around there. And then also their Golgi nuclear axis alignment becomes much more randomized right around that portion. It's almost this nice step function. And again, this behavior is borne out again by their this polarization is borne out by their migration persistence. And so, again, once the cells encounter an amplitude that's around 15 microns or larger, they become less directionally persistent. And I should say that in some data I don't have time to show you today, we don't see a change in the cell speed, just their persistence. So to understand how these straight and small amplitude waves were allowing polarization, we want to inhibit several of the key components of the polarization machinery. So when we inhibit either myosin contraction with plavastatin, or if we inhibit the nucleation, actin nucleation factors, or if we inhibit the important polarization signal PI3 kinase, we see that all of these will somewhat suppress the elongation of the cells and their aspect ratio, particularly at the lower wavelengths and on straight ridges. And so, of course, in the high amplitude ridges, we don't see any effect because the cells are already depolarized. And so, while this isn't very surprising, we wondered, how are these cell components spatially organized to enable cell polarization? And what might the large amplitude wavy bundles be doing to alter this organization to cause depolarization? So when we use the GFP tagged pH domain of AKT to look to see where PIP3 is, we see that PIP3 is, rather than being at the cell edges, it's localized along the ridges on all the substrate types that we tested. And since one of the advantages here of these sort of very regular wave substrates that we're making is that since the waves are all identical, we can average together many half waves and look at the sort of average distribution of molecules on these waves. And what we see is that PIP3 is accumulated at the wave peaks, which is kind of reminiscent of what we might see in a leading edge lamellipodia. So we're kind of now thinking, well, maybe these are sort of like little mini lamellipodia inside the cell. And indeed, when we look at other actin lamellipodial machinery, such as cortactin and ARP23 and free barbed ends, which I'm not showing here, they too are likewise localized and enriched at these wave peaks, as you can see here on the right. So we think that these curved ECM coated ridges create sort of internal lamellipodial-like structures. And since I told you that adhesions are tightly coupled to the lamellipodial actin polymerization, how would we expect the adhesions to be organized? And so before I get to that, I should say that while we are thinking about these as collagen fibrils in terms of how the tumor stroma is organized, at least we find in this in vitro system, at least that the specific matrix component does not matter that much. So whether we coat these substrates with collagen or fibronectin or even a complex mixture like Matrigel, they will all support this polarization on straight and small waves and depolarization on large waves. But if instead we use a sort of a non-specific sort of cell glue like concanavalin A, we don't get the same elongation response, which at least at early times before the cells can make their own matrix. And so this suggests that we need integrin engagement to see this polarization response on the low amplitude and straight fibers. Sort of corroborating this, we also see that if we compare wild type and FAC null fibroblasts, we see that there is some requirement for some contribution of FAC activity to the ability of cells to elongate on the low amplitude and straight ECM fibrils. So we think that then generally the integrin-based adhesions are required for this response. So then how are they organized, right? And so the focal adhesions, similar to the lamellopodial machinery, are also enriched on the wave peaks on the large waves. But here we see something different for the small waves for the first time, which is that instead of being at the wave peak, we find that it's that like the paxillin is shown here is enriched sort of on the shoulders of the wave peak. And so when we look a little closer, we see that instead of sort of making sort of stress fibers that are kind of sort of spanning across in a whole arc, what we see are these little tiny mini stress fibers within some of those smaller ridges. And then alternatively, the second kind of thing we see is that we see long stress fibers that extend across many ridges. So it's as though you can't really fit in a stress fiber or an actin arc in those tiny little curves. And so when we sort of dig a little further into this and we look at the maturation organization, of these focal adhesions, we can use a proxy for the maturation orientation, the distributions of paxillin and zixin. So normally paxillin is found sort of throughout the focal adhesion, but is enriched towards the distal tip of the focal adhesion. And zixin tends to be more in this sort of more proximal region of the focal adhesion near the stress fiber, and then can be found up into the stress fiber. And so when we use these two molecules and look at their orientation, we see that on the straight ridges, that we get this sort of axis of orientation is along the ridges as we might expect. And then on the small wave, we also see where this axis of sort of elongation of the focal adhesion is sort of along the ridge wave. But on the large wave substrates, which are the depolarizing ones, what we find is that this paxillin-zixin axis is actually orthogonal to the wave pattern, wavefront axis. So they're sort of contracting against that wave curvature. And so this is, again, borne out by and consistent with how the actin is organized. So on the straight ECM fibril ridges, we see that the stress fibers are elongated along those ridges, as we might expect. And around the large wave ECM fibrils, what we see is that there the bundles are bent around these large wave ridges, right? And so we see some things sometimes where there'll be some that'll span across it, but generally they're bent around them. And then the weird thing is that on the small one, as I showed you, sometimes we see these very short, small stress fibers that kind of seem to run from peak to peak in the large, or in the small waves rather. But generally, though, when we look a little bit higher in Z, we find that the F-actin and the stress fibers look a lot more like they do on the straight. So this is now correlating their behavior and shape in how their stress fibers are organized. So then what about the myosin? Well, so what does this do to actin myosin organization? So what we find is that on large waves, if you see up here on the upper right, the myosin is contraction field is organized into opposing fields, and it always contracts against the curvature of the wave. So what this sets up in these large wave substrates is these sort of opposing fields of myosin contraction. But in the small wave substrates, we see something a little more reminiscent of how a cell that might elongate and crawl along a fiber axis going left to right here. So we see that the myosin contraction is, again, consistent with the way we saw the stress fiber organization. So the last thing I want to say before I end up here then is that while we found that all cells tested, every cell we tested, whether they're endothelial cells, fibroblasts, tumor cells, what have you, they were all depolarized by large amplitude ECM waves. We noticed that more contractile cell types tended to be less affected by these large waves. And so if we just use the regulatory myosin light chain phosphorylation as a proxy for actin myosin contractility, we see that the more contractile cell types, like the MEFs shown here, tend to elongate more, even at some of these sort of depolarizing wavelengths around 15 microns. And this is also borne out by cancer cell migration. So migration persistence is slightly enhanced at this depolarization, these depolarizing amplitudes in cells that have a higher actin myosin contractility amount. So what we think then is that the intrinsic contractility, actin myosin contractility of cells may modulate how strongly a cell is depolarized by these large ECM waves. So just to sort of wrap up here and think about how does this work. So our model is that the adhesion organization of on these sort of different types of wave structures or ECM fibrils is what drives a lot of this spatial organization. So on straight fibers, on linear ECM fibers, the adhesion and polymerization machinery are organized along this axis of the fibril propagation. And this has been well described by other groups also, such as the Provenzano group. But when the fibers have large waves in them, this supports sort of organization of adhesions and lamellipodia and actin-myosin flow in multiple directions, which might confuse the cell. Conversely, though, when the amplitude is too small, this doesn't support such an organization. Instead, what we think is that these smaller sort of arrangements of the actin polymerization machinery instead just knit together to make longer stress fibers and enable the cell to crawl sort of longitudinally along the wavefront propagation axis. So how would this then affect cell, how might this affect cell behavior in a tumor? Well, in the Caillé sort of very advanced stage where you have these straight roads, we anticipate that this would lead to a cell just trucking right on out of there. But in the case of the sort of more benign stroma, what we anticipate is that it might lead to sort of a more confused behavior. And so the cell might be being given polarization directions that are in multiple directions and then therefore really not get anywhere in general. And so the cell is then confused and can't kind of get out there. And so what we would suggest then is this cage that we see in our skin tumors of wavy collagen fibers might be like a depolarization barrier and prevent cells from getting out. And so I'll just end by thanking Claire, who's been my longtime collaborator and mentor and friend. And I want to give a shout out to Michelle, who's the graduate student in our lab working on that great, really cool melanoma project. Anna and Bill in the lab have been my longtime co-partners there. Bill manages a lot of the microscopes in our group, and Anna's sort of my work wife, as I say. Former lab members have given me a lot of inspiration and thoughts on this subject. Vinay, Val, and Sergey have all gone on to make their fame and fortune elsewhere, running their own labs. Chris Combs helped us do some of the 2-photon imaging. Zhao Yusun and John Forkas made those substrates in the first place and have really enabled this whole project. And then Claudia Fischbach and her former postdoc, Bo Ree, who is now at Harvard, gave us access to their collagen data. And so I'll end there and take any questions.
Awesome. Bob, thanks so much. That was really great. I just want to ask myself 2 questions first, and of course, everybody should drop questions in the chat. So just for clarification, all of the topography is all in plane, right? Like there's no, it's not rolling hills. It's all flat.
Yes, correct.
Okay.
Correct.
Okay.
Correct,
just
correct.
because
Yeah.
I wasn't quite sure from the images. So just to double check. And then I wanted to ask, you mentioned about stiffness in the beginning and thinking about, well, what are the stiffnesses of these materials? Because what I would picture, and I don't know if this is right, but in vivo, I would guess that these wavy ECMs should be a lot softer, right? Because they're not they're not supporting tension.
Right.
So I was just wondering if you could comment a little bit on thinking about that, because that should be, in addition to the shape of the topography, I would imagine would be a difference in vivo.
Yeah, no, for sure. I mean, the stiffness, so these are acrylate, so they're basically infinitely stiff, right? And so on the one hand, that is not very, in vivo like, because we do anticipate that the collagen fibers that are wavy might be, you expect those to be softer. But on the other hand, we kind of want to isolate all the other components of the sort of cell polarization machinery, just to ask the question, sort of in a very reductionist way, does the shape itself contribute to the cell behavior? Ideally, what we'd love to be able to do is to be able to make actual collagen fibers that are those shapes of some defined shape. Instead of doing that, one of the things that I've been playing around with, and it's sort of not ready for prime time yet, and Madeline showed it in some of her work, is using actual tumor ECMs, decellularizing them, and then sort of asking, when the cell encounters sort of this cage of wavy bundles, do they indeed go slower, or do they, become confused? And I think that might be the real direction to go into, rather than trying to somehow, because I can't really figure out how to make these things in 2D that would be compliant and in the specific shape. So we might have to have the tumor tell us, make the things for us. But that's a great question, and it's an important point that we definitely have to keep in mind, that the stiffness is going to matter as well. I think there was maybe some questions.
I can read a few out, Bob.
Okay, yeah.
So Hamid, yeah, I mean, we'll try and get through maybe 2 or 3 now, and then maybe we can come back at the end so we can give Isabelle
right.
First question from Hamid, very similar to what I was going to ask. So how does the transition from wavy to linear happen in vivo?
How? Oh, like you mean? So I guess the question is, well, there's a couple ways. Is the question that how are there transition zones that sort of go from sort of straight to wavy, or how does
I
the
guess it's
micro get reorganized?
Yeah, I'm guessing it's
Okay,
about the
yeah.
reorganization.
So I what I think is that and this is just really based on just the sort of observational sort of things that I think what's happening is that the tumor cells are the ones doing the yanking and making the fibers straighter. I think because what we see when we look at the other cell types that surround these skin tumors anyways, is we see a lot of cells, non-tumor cells around those tumors, and they're in that kind of curvy sort of wavy nest, right? And so I think that the local probably macrophages that are being recruited, as well as the local mesenchymal cells, that may be cancer-associated fibroblasts, are making sort of that curvy bundle, and it's the tumor cells themselves that are probably yanking and pulling and making this more straight matrix. And it could be through a variety of ways. It might not be just mechanical. It might be that they're digesting elastin, for instance. So elastin is one of the things that can help make the fibers wavy, and they might be digesting other things. So they're definitely making some amounts of matrix metalloproteases, things like that. So but I think it's the tumor cells that are doing it. But that's just a guess.
Great. And then I think maybe we do have more questions for you in the chat. You can always answer them
Yeah.
there, or we can do it afterwards. But maybe this will be the last one from Erica Singos. Do cells of different sizes react differently to the different amplitudes?
So that's a great question, and I think I don't have definitive proof. You put a gun to my head, I think yes, they do. Larger cells will require larger wavelengths to depolarize. That's something that Val, our former postdoc, and I talked a lot about. Maybe trying to put like neutrophils or tiny cells to see would they be depolarized differently. I think that the overall amount of myosin contraction the cell can generate kind of regulates that somewhat, but I do think the cell size matters, and why that would be. I can only hazard the guess that it's the ability of the cells to generate sort of fields of adhesion that would span large enough to become depolarized. But that's, just a guess.
Great. Thank you so much. We really appreciate the great talk. That was really interesting work. And like you said, hopefully it'll be appearing soon.
Right. Thank you.
Thanks a lot. And Isabelle, if you want to go ahead and start sharing your screen. So I'm really happy to introduce our second speaker today. We have an awesome double header. So we have Isabelle Caillé. She's an assistant professor at the University of Paris. She was a postdoc in Arturo Alvarez Buya's lab. You can correct me if that was wrong. So he's one of the discoverers of adult neurogenesis, and she has since stayed in the field of adult neurogenesis. She was first interested in the identity of adult neural stem cells, then in the differentiation of their progeny, and ultimately in how young neurons migrate in the postnatal brain, and that will be the topic of her talk today. Your screen looks good. You've got it shared, and
okay. So let
go ahead,
me
take it away.
Have a special mouse here because we couldn't see any of Bob's mouse. So can you? That's it.
Yeah, I can see your regular. Just
my regular mouse is fine. You can see it.
I mean, it's small, but it's there.
Okay, great. So thanks to the organizers to invite me. I feel a little bit exotic here just because maybe I'm French, and also because there's not so many there are not so many talks in your series about neuronal migration, but I can assure you that neurons do migrate, and that they're highly polarized cells. So I will talk to you today about how primary cilium-dependent cAMP-PK signaling at the centrosome regulates neuronal migration, which was recently published in Science Advances. So as you might know, neuronal migration is a crucial step of brain development, since all neurons have to migrate from their germinal niches, from their sites of birth, to their sites of integration, and sometimes quite long distances. So here in the mouse embryo, embryonic brain, you can see that some neurons might have to migrate very long distances, like these ones, and some shorter distances like these ones. And it's the proper migration is absolutely necessary for the brain to build normally. It exists in the embryo. It exists also in the postnatal brain. And here are depicted newly discovered path of migration in the infant brain. But what I will talk mostly about today is migration in the postnatal mouse in this pathway, which is called the rostral migratory stream. So on this sagittal section of the mouse brain, postnatal mouse brain, you can see that the neurogenic niche here is called the subventricular zone, where neural stem cells reside and give birth to very numerous neurons, which migrate along this restricted pathway to this anterior part of the brain, which is called the olfactory bulb, and where they differentiate into inhibitory neurons. But it's a massive migration. It's more than, in the adult, it's more than 10,000 neurons per day, but in the postnatal brain, it's even more massive. So they migrate like all the other neurons in what we call a cyclic saltatory migration, which is an alternation of somal translocation and pauses, and with different steps. So here is a neuron, a young neuron migrating. Here is its nucleus, and here is its centrosome. So it initially starts with a pause, then the leading process elongates, and the centrosome migrates along inside the leading process in a process which we call a centrokinesis. And after that, the nucleus follows the centrosome and performs a nucleokinesis. And then there's another pause, and it starts all over again and again. So pause, centrokinesis, then nucleokinesis, then pause again. And in this system, the neurons, they spend nearly 80% of their time in pause. So it's really nice life they have. So it's also called the two-stroke migration because of these independent events, or nearly independent events, of moving the centrosome first and the nucleus after, which I don't think you have in other types of cells. So another very important protagonist of this story is the primary cilium, that you also might know. So it's a small organelle which is present, which protrudes at the surface of most eukaryotic cells, and including neurons. It is a small sensory organelle which is linked to the centriole, the centrosome, and more particularly to the mother centriole. It contains an axoneme. It's considered to be a hub of signalization, and it's essential for brain development, as evidenced by the very severe brain defects in human ciliopathies. So it's important for many steps of brain development, and including neuronal migration, which was shown mainly by two important papers in the field. One from Eva Anton's lab, and the other one from Christine Métain's lab, in which they showed that the primary cilium is fundamental for neuronal migration, since when you ablate it in a mutant, in a ciliary mutant, this pathway of migration here, the long migration I showed you in the first scheme, instead of having neurons all over there, those neurons are severely delayed. So then, however, it is important, but we still absolutely don't know how primary cilium signaling can be converted into a migratory response. Still, we know that the primary cilium is a cyclic AMP, very rich source by different, it was shown in different papers. So we wondered whether, this is a pain, the PC, the primary cilium, could regulate migration through CMP signal. So to do that, we used a CMP-specific FRET biosensor that we live imaged. So it's schematized here. When there's no CMP, the biosensor is in FRET conformation and thus emits at this wavelength. Whereas when there is CMP, the biosensor is in non-FRET conformation and thus emits at this wavelength. So we measure the emission at both wavelengths, and at each time point of each pixel of the movies I will present to you, we make the ratio of this wavelength, or the intensity of signals of this wavelength over this one. And it will be represented as false colors, with red being high concentration of CMP. So what we do is that we electroporate. So we don't use cells in vitro. We electroporate a neonate mouse. So we inject the plasmid encoding the FRET biosensor in the lateral ventricle, the cavity of the brain. And then we put the electrodes and we electroporate the mouse so that, and then it recovers, so that it transfects the cells which are lining the ventricles here, which are the neural stem cells, which will give rise to the migrating neurons, which will then express the biosensor themselves. But all the experiments in what I will present to you are done this way by postnatal electroporation of young mice. So at P2 postnatal 2, we electroporate, and at P7 to P10, we perform, we do acute sagittal brain slices, and then we perform 2-photon live imaging and ratiometric analysis to analyze CMP dynamics. So what we saw was this type of movies. So this is a migrating neurons in the RMS, in the pathway of migration I talked to you about. And as you can see here, you see a neuron performing 2 successive nucleokinesis. And again, so, and you can see that there's a red hotspot transiently appearing in front of the nucleus here, and then again, and then disappearing. So there's kind of a blinking hotspot. We were very surprised to see that. By the way, the biosensor is not addressed anywhere, is not targeted anywhere in the cell, it's just all over. And also it does not enter the primary cilium. But so if now we look at still images of the second nucleokinesis here, we can see that here, there's a red hotspot. It's visible here, 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 and here. And before this time and after this time, it would be absent. So as this movement made us think of the movement of the centrosome during migration, we co-electroporated the biosensor with a marker of the centrosome, a centrin RFP. And what we could see was that, so here on the left panels, it's the biosensor, and you can see very, very nice hotspot here, here, and here. And here, this is with the confocal imaging of the centrin RFP 1 minute later. And so you can see the centrosome, and you can see this perfect localization. Yeah, nearly perfect, since there's a 1 minute delay. So we can conclude from this that there's a dynamic CMP hotspot at the centrosome during nucleokinesis, as schematized here. So during the pause, nothing, and then something appears here at the centrosome, and then it stays during the whole nucleokinesis, and then it disappears. And this was present in more than 80% of the cells, as seen in more than 100 cells in 10 different animals, by the way. So here is a heat map of the frequency of occurrence of the hotspot in 14 neurons performing nucleokinesis, with the 0 time being the middle of the nucleokinesis. So you can see that how the hotspot is frequent. So then, of course, as the primary organelle, and it's just above the centrosome, we wondered whether the hotspot we saw at the centrosome could emanate from the primary cilium. So first we asked whether those migrating neurons had a primary cilium, and this was indeed the case, since as you can see here, this is a nice GFP labeled, also electroporated, same technique. So plasmid with GFP was electroporated, so it labels a cohort of new neurons, newly produced neurons migrating in the RMS, and then a fixation of the brain of the animal sections of the brain and immuno with both gamma tubulin to label the centrosome and Al13B to label the primary cilium. And you can see that there is a primary cilium in some of the migrating cells. You can see it's full of neighbors here, non-GFP labeled neighbors, which also have cilium, as you can see. So then we to understand if the primary cilium was important for the production of this hotspot, we used 2 different fluxed mouse lines in which 2 different genes were fluxed, either KI3A, which is necessary for the establishment of the primary cilium, maintenance of the primary cilium, and RP1L, which is another gene which is necessary for in these cells for the primary cilium to be present, so that when we electroporate the Cre recombinase in a plasmid in the lateral ventricle along with the biosensor. So when we electroporate the Cre, those genes are recombined, and so we perform a primary cilium ablation. This was verified, of course, by counting that. And then if we do that and co-electroporate the biosensor, what we see is that, meaning nothing. I mean, the biosensor is expressed, but there's no hotspots. The hotspot has disappeared, and this was, of course, counted. So this is the situation I already showed you for the wild type, with the vast majorities of the migrating cells harboring a hotspot, a periodic hotspot. And those in the KI3A line recombined, it was reduced, this percentage, and even more reduced in the other cell line. I mean mouse line. So the primary cilium seems to be necessary for the hotspot formation. So we wondered how. And the main cAMP producer in the primary cilium of neurons is the adenylate cyclase 3. And it's membrane bound at the level of the primary cilium usually. So that's what we verified in our cells, and that's what you can see here with a nice, with an immuno of a newly formed migrating cell. Here it is labeled by GFP, and it's also labeled by Al13B, which labels the primary cilium, which is tiny. And it's co-labeled with AC3, and we quantified this and saw that, oh no, on AC3 and CB effects on the presence or not of the hotspot. And we could see that, yes, indeed, knocking down AC3 triggered nice disappearance of the hotspot. So we show that the primary cilium produces the hotspot via AC3. So then we wondered whether this hotspot could have an influence on neuronal migration. And to do this, we performed a series of experiments using the KIF3A LOX mice. And we electroporate them either with GFP to have control, or with CRE-GFP to ablate the primary cilium, or with the MIR-AC3 to ablate AC3, to knock down AC3. And then we perform confocal imaging between P7 and P10. So this is an example of a GFP electroporation. Here is the neurogenic niche. Here is the pathway of migration. And here is the olfactory bulb. So we typically film those neurons here in this pathway. So what the kind of films we make are like this. So you can see that we have this cohort of GFP positive cells nicely migrating and performing, as you can see, regular saltatory migration with elongation and then moving, and then first elongation of the leading process and then nucleokinesis. Here you cannot see the centrosome because it's not labeled. And this is a control. And at the same rhythm, you can see the pre-electroporated mice. So this is supposed to be the same rhythm, but you can see that the neurons, they do migrate, right? They manage to migrate, I have to say. And ultimately, if you wait long enough, they will reach the olfactory bulb. But their migration seems to be much more difficult and slower. And they're even more in pauses. So of course, we quantified all this. So we measured the speed of migration and could see that it was indeed reduced in the two types of mutants. So as a reminder, CRE-electroporation to ablate primary cilium, MIR-AC3 to knock down AC3, and triggering the absence of hotspot. So in the two types of mutants, the speed of migration is reduced and the pausing time is augmented. And this is accompanied by a reduction in the frequency of nucleokinesis. So from this, we can infer that probably the hotspot regulates the rhythmicity of migration. We wondered then if it could be an effect on the centrosome, since the hotspot is typically at the level of the transiently on the centrosome. Could it act there? And so to assess the centrosome movement, we co-electroporated GFP with centrin RFP to perform confocal imaging. And here it's also on a slice, and you can see it's very nice movement, like in the scheme. So first the centrosome moving and then the nucleus moving repeatedly. So what we did was either to electroporate GFP and centrin RFP, or CRE and centrin RFP of MIR-AC3 and centrin RFP. And we could see that the nucleus to centrosome maximum distance was reduced in our mutants. And also we measured the efficiency of centrokinesis. So officially the centrosome should move first, and then in this swelling, it should move first, and then the nucleus follows like this. And this is what we call efficient centrokinesis. But we also can see some inefficient centrokinesis, which would be the centrosome moving far enough, but then the nucleus not following. And we could see that the centrokinesis efficiency was reduced in our mutants. So from this, we can conclude that the hotspot seems to regulate centrosome to nucleus coupling. So we wondered what could be the downstream effector of the hotspot. So there's a CMP enrichment at the level of the centrosome. On what could it act at this level? So it is known that in certain types of cells, including some neural cells, that the cAMP-dependent protein kinase A is localized, is subcellularly localized at the centrosome. So we verified if it was as a reminder, so the pKa is formed like this. It's for 2 regulatory subunits and 2 catalytic subunits. And when cAMP is bound to the regulatory subunits, it releases the catalytic subunits, which can go and phosphorylate whatever target they want. So we verified whether pKa was localized at the centrosome of our cells, and it was really the case. As you can see here, it's an immuno against the catalytic subunits, and it appears that here as a blue cloud, and you can see that this blue cloud is surrounding gamma tubulin here, including in a newly formed GFP positive. So then we wondered what would happen if pKa wasn't localized at the centrosome, and we asked a plasmid to perform this experiment to Elisa Marti's lab. It's a dominant negative form of pKa in which the regulatory subunit cannot anchor the centrosome. So that it traps the endogenous catalytic subunit outside of the centrosome in the cell. It delocalizes by immuno, and you can see this nice little cloud here, and which typically are close to a centrosome, except for this cell, which has been electroporated with this DNPKA coupled to the GFP. And in this cell here, this is the white arrow, there's no blue cloud. So the pKa is not at the centrosome anymore. And we counted it, and you could see that the percentage of cells of migrating cells with no pKa at the centrosome was indeed very much increased. And of course, we analyzed the migration of these cells with delocalized pKa and could see that their speed was reduced, the percentage of pausing time was increased, and the nucleokinesis frequency was reduced as well. Again, we analyzed the centrosome movement and could see that the nucleus to centrosome maximum distance was reduced, whereas the centrokinesis efficiency was decreased. So as you can see, it really nicely phenocopies the other migration. It phenocopies cilium ablation and AC3 knockdown. And in addition, there's no additivity of the phenotype in the double mutant in which we perform cilium ablation plus DNPKA, suggesting that it's really in the same pathway and that the centrosomal PKA is the downstream effector of the hotspot. So to finish the story, we wondered whether this could be a general phenomenon in migrating neurons and analyze the presence of hotspot in different kind of neuron. It was indeed the case in radially migrating postnatal neurons, as you can see here, this big hotspot. It was also existent in the adult brain, where some neurons still migrate in the adult RMS. And it was also the case in the embryonic brain, as you can see in these neurons. So as a conclusion, we can say that what we observe is a general feature of migrating neurons. And we can say that we have discovered a new mechanism regulating neuronal migration. This is a giant primary cilium with AC3 here at the membrane. And in red is the concentration of cAMP. Here is the pKa and the centriole. So the primary cilium of migrating neuron produces cAMP through AC3. AC3 produced ciliary cAMP is visible at the centrosome as a dynamic hotspot. And this cAMP hotspot acts on centrosomal PKA to regulate migration through nucleus centrosome coupling. So in this, we can say that the primary cilium is a regulator of the rhythm of neuronal migration. And this is very interesting in the context of a recently published paper from the Sawamoto's lab, in which they analyzed the primary cilium dynamic during neuronal migration in the same neurons. And they can see that the primary cilium is emerged only partially during migration. So during pause, you have a kind of incarnated primary cilium here, and it emerges before and during nucleokinesis to be re-immersed in the cell during the following pause. So if you remember well, the emergence of the primary cilium seems to coincide with the hotspot appearance. So it leads us to think that the cyclicity of the cilium could induce the cyclicity of the hotspots through AC3 activation by an as yet unidentified extracellular signal. And this induces the cyclicity of migration. So that's it. And I want to thank all of the lab for this work and the collaborators and the platform. It was a very nice collaboration with Pierre Vincent, and it was mostly performed by two great PhD students, Julie Stoufflet, who's now a postdoc in Laurent Nguyen's lab, and Maxime Cholet, who's now doing something else. And I guess that's it. So I'm open to questions if we still have time.
Yeah, absolutely. If you don't mind staying a few minutes over, and we started a few minutes late for you. Thank you, Isabelle. That was really, really interesting. And as you said, it's a different model system than a lot of us who think about cell migration are used to thinking about. And I'm really fascinated by the idea of the primary cilium emerging as sort of a sensor. Do you have, I know you sort of ended with that idea, but do you have any speculation about what, because something, if that's really the regulator, right? Something's telling it, okay, it's time. And so do you have any idea what that could be?
Well, we're testing different kind of ligands, but it has to be an ubiquitous ligand, since it seems to be in all migrating neurons. So we thought of different things, and I would very much like something like a pressure to be that trigger, but we didn't find any. We have to go further. We thought of the pressure, we thought of the pH, something so or mechanical, or just the quantity of salt outside. Or it can be a specific ligand of a GPCR present on the primary cilium. There are different kinds. So we thought of GABA, since these neurons are GABAergic, but we are now, we tend to think it could be another type of ligand now. So.
Yeah, and I think in other systems, I don't know about this one, but other systems, the primary cilium can act as sort of a flow detector, right? So is there interstitial flow that it could be detecting or responding to?
I would very much like it, but I don't know exactly how to test it for now. And it should be a GPCR on the primary cilium coupled to GS. That would be the best candidate. It should be on the primary cilium and activate GS.
Okay, fascinating.
And send it to the flow. So in case you have any idea, I buy it.
Yeah, no, I think this is a time for a literature deep dive, because I've heard of these things, but I don't know. Let me switch over to YouTube. We have a question on here, which of course for some reason I can't quite see. Okay, from Qureshi Haroun, asks, during live imaging, how do you actually assign specific time stages of migration? They're assuming that there may be some random pattern that's also appearing during the migration. So how do you filter that away?
So we take a picture every 3 minutes, and the threshold for it being migrating or not being in pause, as opposed to pause, is moving more than 6 micrometer. So that's a threshold which is used by others in the literature.
I've got a couple of questions myself, which maybe I can ask. I was wondering, So even when this is defective, you showed that you mentioned that the cells kind of struggle to, but eventually get there in the end. Yeah. So is there any long-term defects that, so like maybe the total number of cells that reach the end ends up being lower, or something that, or at the very end of it, would it be the same?
At the end, from what we counted, at the end of it, all of them finish. We have, in the longer term, we have the regular amount of neurons, but it doesn't mean that it's not. It doesn't create defects, because the timing of arrival also has an importance.
Okay, so the rhythmicity of that migration is really important to prevent defects.
You mean migration to the factorable? Yeah. Yes, but also we showed that there are defects in the migration to the neocortex, which is even more important. This having enough inhibitory neurons in the cortex is really of paramount importance to a regular functioning. It's and this disbalance between inhibition and excitation is involved in a lot of psychiatric diseases.
Yeah, and my other question was kind of related to what Jen was kind of touching upon about what the activator was upstream of the adenylates. So I was wondering, but I'm not sure if maybe you did this, remember I missed it, or maybe it's not possible, about whether you can constitutively activate that to extend the nucleokinesis stage and see whether you can.
By doing what?
In order to increase the time in which it's
but how to activate? Just by
oh, I have no idea. I was just asking.
It would be possible just by using a regular pharmacology for cAMP and using but it's very artificial. I would rather find the regular ligand, the normal ligand or the normal receptor involved, and then knock it down.
Of course, yeah.
So I have to find it first.
And then another question, we've got one from Raphael, but I'm going to ask one for myself again first. Is there any sense of a mechanical regulation? So one thing that I was noticing is that it's sort of, it looks like the protrusion gets to a certain length and then it's almost like a snapping, right? Where the nucleus is sort of being rapidly pulled forward. Is there any evidence that it could be sort of, it's as if you've stretched a rubber band to a certain length?
Right.
And then and then that's a mechanical trigger saying, okay, it's, now we pull it forward.
I think there's a limit. So I was trying to read some things about the forces before talking to you, because I was sure you would ask.
I'm very predictable.
And I don't know much about it. But, this is forces to drive neuronal migration steps. And yes, some people are trying to measure the tension, and there are some schemes with some springs and the forces, and of course there is a max. There is something with the tension which can ultimately trigger the movement, but there's also actomyosin at the back pushing.
Oh, okay.
So
it's not just a pull, it's a push and a pull.
It's a pull and a push. And that might be the reason why they're ultimately, they still move anyway, because they're still pushed.
Okay, great. Yeah, when I was in grad school, I worked with Schwann cells, and they would do the same kind of migration where they'd be like unipolar and then snap to the front.
Right. But in other types of cells, you don't have that, right? It's only in neurons and neural cells.
Yeah. I mean, I personally saw it in my experiments with these glial cells. I think I've seen it in the literature a bit, but
no, yeah.
Yeah. So it's really, really interesting. So we have a question from Raphael Petrosian. Thanks for a very interesting talk. He says, how fast can neurons migrate, and what are the defining factors of their migration speeds?
So from what I showed, what was the average speed here was, let me look back at the speed I was describing before. I'm looking on my computer. I think they're around 70 to 80 micrometer per hours. And so what defines the speed?
That's fast. I'm impressed. That's quite fast. I think that's fast. I don't know what other people think. I think that's fast. It's not maybe not fast compared to like rolling neutrophils or something, but it's fast compared to epithelial cells that I work with.
What's your
Yeah,
speed?
just don't ask the immunologists, because then they'll say no, that's not fast. But it's
Right,
fast
right, right.
for any non-immunological cell.
For any crawling cell, I think
Correct.
it's fast. Yeah.
Oh, I see. So we are the speed here, yeah, was around 75. Yeah, that's it. I'm not saying bad things. So and what defines the speed? Well, the speed is defined by the fact that all the intracellular processes are going well, which means all the movement of the centrosome, the nucleus, and everything. And I don't know what the question means exactly, I guess.
No, I think that's great. Thanks. And then we have another one from YouTube. Diego Garcia Gonzalez says, "Nice talk, Isabelle. Do you expect any difference between the radial versus tangential neuroblast migration if the primary cilium is more prominent in neuroblasts in the OB?"
So Diego is a neuroscience person. I know him.
Must be.
Do I expect difference in terms of the role of the primary cilium in migration? Is it?
Yeah, radial versus tangential neuroblast migration.
And do I expect the primary cilium to have a different role or not? What is the question exactly?
Yeah. Do you expect any difference between radial versus tangential neuroblast migration if the primary cilium is more prominent in neuroblasts in the OB?
He's talking about tangential and radial migration along the same system. And what we do see is that the hotspot is present during the radial migration, but the dynamic is not entirely the same. And also we saw that the primary cilium is longer and more present during radial migration. So there are some differences, even if the mechanism is conserved in the two directions.
Okay, great.
Maybe we should talk directly.
Yeah, I'm sure he can reach out to you. And there's a delay on YouTube, so he'll get your answer in about 30 seconds, and we'll see if that's
okay.
All right. I think that's it for questions. So I just want to say thank you to both Bob and Isabelle for really lovely talks on different topics, but also migration. So it was really great. And then I'm not sure if the folks who had asked questions are still around, but there were a few that we didn't get to for Bob's. So if you guys want to try to follow, we can try to follow up on some of those real fast at the end here.
Okay.
I've lost them in the chat, and I'm not sure which ones specifically you already answered.
I tried to answer the longer questions, so I think it's probably, but if
Sorry, Bob, I will say goodbye to everybody because there's a curfew here in France and I want to buy bread.
Oh.
Yes. Oh, that is more important than any of this. Get your bread. Absolutely.
Yeah, go buy bread.
Sorry. Bye.
Great talk. Great talk.
Bye. Thanks.
So nice to meet you and great talk. Thanks, Isabelle.
Yeah,
why don't we? Yeah. Oh, bread's more important than anything. Why don't we just give it like a few seconds if anyone here wants to ask their question to Bob? Otherwise, we'll say goodbye. All right. I think we're losing folks and people got places to be. So
that's right. Thank you guys so much again for the invite.
All right. And if you ever figure out how to turn on the black and white filter, email me, let me know so that I can learn how to do it because it looks super cool.
Yeah.
Okay, I will.
It's very classy.
All right.
Very classy. All right. Thanks again. That was really interesting. And
sure.
We'll
Thanks,
see you again
guys.
soon.
All right. Bye.
Okay. Bye.
Bye, everyone. See you next week.